Welcome to KayScience's third instalment of the online tuition that we're offering for free. Let's just go through quickly what KayScience offers you. So you can click on any of these videos, and if you don't understand the topics or you've forgotten it, you can watch the videos, and then at the end, you've got question time, and then you've got gap fill exercises, and then you've got exam style questions at the end of every video, as you can see right here. And then the answers are shown directly afterwards. So you can click on any of these videos and you can go between them very easily. Then you've got the quizzes below um, where you can test your understanding of what you saw in the video and then mark live. So if you write the incorrect answer, it will correct it for you. And then all of these quiz scores are tracked here. So you see what you can do and what you can't do. And that's for biology, chemistry, and physics. And this will, if you watch all of our videos and attempt all of these quizzes, guarantee you grade 8 or 9. That is if you get all of these questions right and watch all of our videos. So let's begin. Let's now do some revision on enzymes. So enzymes is a really important topic. So we're going to start off with the lock and key theory of enzymes. This here is an enzyme. This here is an enzyme. And the enzyme breaks down substrates into smaller products, or it makes larger products from smaller substrates. In this example, we're going to show the breakdown of a substrate into smaller products. So this here is a substrate. The substrate is going to bind the active site of the enzyme. So this here is the substrate, and this is going to bind the active site of the enzyme. As you can see, these substrate shape and the enzyme's active site shape are complementary and they are specific to one another. So what happens is the enzyme is bound by the substrate, and that happens in the active site. So the substrate binds the active site of the enzyme. And the enzyme is what we call the lock, and the substrate is what we call the key. This is why it's called the lock and key theory, because the substrate that binds the active site of the enzyme is specific to the enzyme, just like a lock and a key. So if we show you this example here, this substrate is not complementary to this active site's, so this enzyme's active site. So the substrate will not bind the enzyme because the substrate is not specific to the active site of the enzyme. So it will not bind. So the substrate must be specific to the active site of the enzyme. So let's go back to our example up here. So we've got the enzyme, the substrate has bound the active site of the enzyme, which I'm going to show you now. The active site is bound by the substrate. So this here is the enzyme substrate complex. Why is it called the enzyme substrate complex? This is because the enzyme is bound by the substrate, the enzyme substrate complex. So this is the enzyme substrate complex when the enzyme has been bound by the active site. Sorry, when the enzyme's active site has been bound by the substrate. And in this case, the enzyme then going to break down the substrate into products. So the substrate is broken down into products. You can see that here. These are the products. So products, what word do you recognize from this? What is produced? So what is produced? This is the product. And in this case, the active site was bound by a larger substrate, which then broke down the larger substrate into smaller products. But this can also be the other way around. So let's now do question time. So the enzyme is the, press the answer in the chat, is the lock. An enzyme, is it a biological substrate? I forgot to say this, is a biological catalyst or is it a biological reaction? So I left this out, so it's a catalyst. It speeds up the rate of reaction without being used up itself. Okay, and it's biological because it happens in biological organisms. So an enzyme is a biological catalyst. Okay, what's the substrate bind? 1, 2, or 3? So the substrate binds to the active site of the enzyme. Right, so this here is the gap fill that you get at the end of every video. We're not going to do that now. You can pause it here if you're watching it, and these here are the answers. So you can pause here if you're watching this on catch up. But what I do want you to do is spend 1 minute answering question 1 to question 8. And if you complete questions 1 to 8, just do question 9. And here are the answers.